In this video, I'm going to talk about translation. If you recall the central dogma of molecular biology, which is DNA goes to RNA, goes to protein, and you recall that proteins are the structural components that make a cell work, you'll understand the importance of the second part of this process, which takes mRNA and turns it into protein so that the cell can use these proteins for its various processes. You might ask yourself, why do we call this process translation? Why do we say that changing RNA into protein is translation? And the reason we call it translation is that mRNA is made of nucleotides. So here's a simple diagram of RNA. You've got a sugar phosphate backbone, phosphate ribose, phosphate ribose, phosphate ribose. And onto each of these backbone components, you have nitrogenous base. So here we have an adenine, a uracil, and a guanine. So these are nucleotides that are stacked together to make an mRNA molecule. And by convention, we'll look at this molecule as going from 5' to 3'. These are just facts that you already know that you should recall. So mRNA is made of nucleotides, but proteins, on the other hand, are made of amino acids. So the processes that we've already learned for copying nucleotide structures that we learned in both DNA replication and that we learned in DNA transcription no longer work when we're talking about changing mRNA into proteins. And the reason is that proteins are made of amino acids. And so here you have a structure of amino acids, and you see amino acids have a backbone that's made of nitrogens and carbons. So you have an amine, a carbon, a carboxyl, an amine, a carbon, a carboxyl, an amine, a carbon, and a carboxyl. And because of this backbone that makes up an amino acid, we call this end the end term, or the amino terminus, the amino end of the protein. These are all terms to refer to the left side of this molecule. And the right side we can call the C term, the carboxyl terminus, or the carboxyl end of the protein. Now, one similarity that proteins have with mRNAs is that this repeating structure has one varying part on each side. So we have what we call a methionine, an alanine, and an isoleucine, which bonds to each of these carbons. And these are what we learned about earlier in the term that we called R groups. So methionine is one R group, alanine is another R group, and isoleucine is another R group. So we call the process translation because we need to take these codes that make up mRNA, these nucleotides that make up mRNA, and translate them into proteins made of amino acids. So when we consider this, we know already that mRNA has all of the instructions that we need for making these proteins. But when we look at these letters, we really have no idea how to translate these, how to turn them into amino acids. So let me give you a few pieces of information that will help you translate them into amino acids. First, we're going to call 3 nucleotides in a row a codon. So any 3 nucleotides in a row here will be called a codon. But I'll give you a hint and show you that we'll start at 3 special nucleotides in the order A, U, G. So we will call A, U, G a start codon. So whenever you see A, U, G, that means we're going to start a protein there. Once you know where a protein starts, you'll know that every group of 3 after that, CAA, GUA, UAG, are also a codon. So once you have your start, you'll be able to find these sets of 3 that follow it. So 3 nucleotides in a row make a codon, and what these codons do is 3 nucleotides code for 1 amino acid. So you might ask yourself, why do we have this system? Why do we go from nucleotides, where we have 4 different nucleotides, to amino acids, and we use 3 nucleotides in a row of 4 different options to make a sequence of amino acids? And so you can answer this by doing a little simple mathematical equation. So we have 4 possible bases of nucleotides. We have 4 possible nucleotides. We have U, A, G, and C. So if we made each nucleotide stand for 1 amino acid, we could have 4 amino acids that we code for because they would directly translate. You could have a U amino acid, you could have an A amino acid, you could have a G amino acid, you could have a C amino acid. Now this becomes a problem when you consider that there are approximately 20 common amino acids that make up our proteins. So using one-letter codes are not enough to turn a mRNA into a protein. So let's step up a notch. Maybe let's do a 2-letter codon. So if we do a 2-letter codon, now remember that each letter has 4 options. So we now have 4 times 4 possibilities of codes, which gives us 16 different amino acids. So among these 16s, we might have UA being one amino acid, UG being another amino acid, UC being another amino acid, and so on and so on and so on. That's still a problem though, because 16 amino acids doesn't add up to enough to give us the 20 common amino acids that we know of. And when I say there are 20 common, there are a handful more. So 16 is really not going to cut it. And so what we find when we get to 3-letter codons, when we make a codon with 3 letters, we now have 4 times 4 times 4. We have 64 different combinations of nucleotides. CUA, UAG, AGC, etc., etc. And among those 64 combinations, we have plenty of spots to slot in all of these 20 common amino acids, as well as a few special amino acids, a few special signals that we need for translation of mRNA into proteins. Now, you can figure out how to translate a sequence like this into a protein using a codon chart. And I didn't want to draw a codon chart into this presentation, but a codon chart presents you in a nice succinct manner all of the possible combinations, all 64 combinations of 3-letter codons, and what each combination refers to as an amino acid. So by using that chart, you can take AUG and decide that that's a start codon, and it also codes for methionine. And so you can go down the line and figure out what each one of these codons' amino acids are. So that's nice if you want to use that codon chart to figure out what the mRNA you're looking at codes for, what kind of protein it makes. But let's talk about how this actually works inside of the cell. How does the cell take an mRNA and turn it into a protein? And surprisingly, it's quite similar to the way that you would look at the sequence and turn it into a protein, although it's utilizing other components within the cell to make this happen. So most importantly, you should recall that translation occurs in the ribosome. So the mRNA, though it's transcribed in the nucleus from DNA to RNA, that mRNA is going to leave the nucleus and go out into the cytoplasm, where the mRNA will actually bind with a ribosome. So the ribosome, you can see, has a small unit and a large unit, and the mRNA is first going to bind to the small unit. Now when it binds to this small unit, you'll see that the nucleotides that we've been talking about are all exposed onto the amino acid, onto the mRNA, and I haven't written them all out here, but you can see we've got some AUG, CAA, GUA. So these bind onto the small subunit. The large subunit of the ribosome then sits down on top of the small subunit. And the importance of the large subunit is that instead of binding mRNA, the large subunit actually binds tRNA, or transfer RNA. Now, tRNA is a special structure that's found within the cytoplasm that is also made of RNA, and it forms this cross shape that's really easy for you to visualize. So it's a cross shape. At the top of the cross, an amino acid binds. So here we have valine binding to the top of this tRNA. At the bottom of the tRNA, there are 3 bases exposed. For example, in this one, it's CAU. And we call these 3 exposed bases the anticodon. We call them the anticodon because they bind to a codon. So this is the unit that actually does the translation. It has RNA nucleotides at one end and protein amino acids at the other end. So you can see here on our first step, we have AUG on our mRNA, which binds the anticodon UAC because of our base pairing rules. And this UAC is attached to a methionine. So that methionine is pulled into the ribosome, binds to the mRNA, and we then have methionine in this site of the ribosome. As we proceed, another tRNA will come into the ribosome. This tRNA now has the anticodon GUU, which binds to our codon CAA and happens to bring the amino acid glutamine into the ribosome. Now, through a series of biochemical reactions that you don't need to worry about too much, the methionine will then bind directly to the glutamine to form a peptide bond, and that will create a new polypeptide or a dipeptide because it'll be two amino acids linked together. Once that peptide bond is formed, the entire molecule shifts to the right. So the AUG will shift over, the CAA will shift over, and will now bring GUA into this site. Once the GUA is in this site, the next tRNA will come in. GUA will associate with CAU, bringing a valine into the site. This valine will then bind to the glutamine, and now you'll have a 3 amino acid structure. And this continues on and on. Each tRNA is brought into the ribosome in turn, binds with the polypeptide that is forming, and then the process continues as you move down the line of the amino acid. Now, to look at this in a little more detail, there are 3 stages of translation that we want to talk about. The first stage is initiation. And so if we look at our mRNA sequence and our ribosome here in yellow, the first thing that needs to happen is that our protein translation needs to be initiated. And it's initiated by this codon that I've already told you about, AUG, which is the start codon. So the ribosome will bind the mRNA. The mRNA will then find the codon, or bind with the anticodon UAC, which brings methionine into the ribosome, and that's going to set the start site of protein translation. So there could be nucleotides prior to AUG. There are certainly nucleotides after AUG, but AUG is the codon which will initiate translation of the protein. So once initiation occurs, the next major step that happens in translation is elongation. And that's what I told you about in the previous slide, where each tRNA in turn will come into the ribosome. It will form a peptide bond with the previous amino acid that was already in the ribosome. And this protein will get longer and longer as the ribosome continues to move down the mRNA from the 5' end to the 3' end. Finally, you'll get to termination. I left out an N there. Termination is where you get to a codon that signals the ribosome to stop. So in this case, UAG is called a stop codon. When the sequence gets to UAG, a special termination factor will bind to the mRNA and cause the ribosome to release the growing protein, and you'll have a completed protein. You'll have gone through all of the steps of translation to take an mRNA and turn it into a protein. So these stages are initiation, elongation, and then termination, 1, 2, and 3. The biochemical processes that occur in translation that allow it to all happen are somewhat complex and not necessary for you to understand in this class right now. What I do want you to understand, and here's our overview of translation, is that translation is the process of taking RNA and turning it into protein. So translation turns RNA into protein. That's opposite of transcription, which turns DNA into RNA. And so those two processes are tied together. You'll often hear people say transcription and translation, because that's the full process. Transcription of DNA to RNA, and then translation of RNA to protein. The important things to know about translation are that you have mRNA nucleotides, and when you put 3 of them together, that's called a codon. Each codon is going to relate to an amino acid. So each set of 3 mRNA nucleotides is going to relate to an amino acid. In the cell, these codons match up with an anticodon. The anticodons are found on tRNA, transfer RNAs. And the transfer RNAs have an anticodon that matches a codon, and they bind an amino acid at the opposite end. So the tRNAs are actually what's causing the translation between mRNA and the protein, because they can bind both mRNA and an amino acid. When this is all happening in the ribosome, you have a 3-step process. You first have initiation. That's where the start codon, which is AUG, will bring a methionine and get the whole process started. After initiation, you'll follow it with elongation, and elongation is going to take the longest of all of these steps, because elongation is going to include the addition of a tRNA into the ribosome and your formation of a peptide bond between 2 amino acids to cause the amino acid to get longer and longer, the polypeptide to get longer and longer. So elongation is going to occur on and on and on until you get to the final codon in a protein known as the stop codon. When you get to the stop codon, termination factors are going to enter the ribosome and cause the protein to be released. So initiation to start, elongation for most of the work of making a protein, and most of the work of translation, and termination when your protein is finally completed and then released. So RNA goes to protein in translation, codons relate to amino acids, and these happen by initiating translation, elongating the sequence, and finally ending translation when they make it to the final stretch of an mRNA and a protein has been completely made.